Welcome to the deep dive. Today, we're getting into something really fundamental. Basically, how your brain cells are set up to work, the resting membrane potential in neurons.
Yes, right.
We got some great questions from you all about this. And well, our mission today is to unpack this core neuroscience idea and show why this resting state is actually so dynamic and important.
Exactly. So the rest in membrane potential, it's essentially this difference in electrical charge across a neuron's membrane,
Hmm.
but specifically when it's not firing off a signal.
Okay.
Think of it like potential energy, maybe like water stored up behind a dam. It's got the potential to do work, it's just waiting. And this electrical potential is absolutely critical for neurons to talk to each other.
Right, that readiness. So what actually builds up that charge difference? Which, like, chemicals or ions are involved?
Okay, so the main players are ions,
Yeah.
specifically sodium, which we write as Na plus ion, and potassium, K plus ion.
Sodium and potassium, got it.
And the key thing is they're not evenly spread out. At rest, you find a lot more sodium ions outside the neuron and quite a bit more potassium inside. This uneven setup is really the foundation.
Okay, outside sodium, inside potassium. But how does the neuron keep it that way? It feels like things would just balance out naturally, wouldn't they? It can't just be passive.
Oh, definitely not passive. The neuron's membrane, its outer skin, is selectively permeable. It's picky about what it lets through.
Selective, okay.
And crucially, when a neuron is resting, this membrane is much more leaky to potassium ions than to sodium ions. There are specific channels, like little doorways, and more potassium ones are open at rest.
So potassium can get out more easily.
Precisely. And because there's more potassium inside to begin with,
Yeah.
it tends to drift out of the cell just following its concentration gradient, trying to spread out.
Like letting air out of a balloon, sort of?
Kind of, yeah. And since potassium ions carry a positive charge, when they leave, they take positive charge with them. This makes the inside of the neuron become more negative compared to the outside. That's really the heart of the resting potential.
Okay, that makes sense. Potassium leaks out, making the inside negative. But what stops all the potassium leaving, or sodium leaking in eventually? Doesn't it need active maintenance? A
Well,
pump thing, right?
exactly, you're right on track. That's where the sodium-potassium pump comes in. It's an active transporter, meaning it needs energy.
Ah, ATP.
Yep. It uses the cell's energy currency, ATP, to actively pump ions against their natural flow. For every cycle, it kicks 3 sodium ions out of the neuron and pulls 2 potassium ions in,
3 sodium out, 2 potassium in, so it's constantly fighting that leakiness we just talked about.
constantly. It's working nonstop to maintain those concentration differences. High sodium outside, high potassium inside, and that contributes directly to keeping the inside negative. Think about something like a muscle cramp. Sometimes those issues are linked to imbalances in these very ions affecting nerve signals. It shows how vital this pump's job is.
Wow, okay. So is this resting potential like exactly the same voltage in every single neuron, or does it vary?
That's a good question. While the basic mechanism is the same, the precise voltage, usually around -70 millivolts, but it can vary a bit. It depends on the specific mix of ion channels that particular type of neuron has. So slight differences, yes.
Okay, slight variations. Now, what happens if something messes this up?
Hmm.
If that delicate balance gets thrown off?
Well, if the resting membrane potential is disturbed significantly, the neuron can't function properly. It can't reliably generate or send signals. It's a bit like trying to use a phone with a really unstable battery. It just won't work right.
So it can't fire properly.
Right. This disruption is actually the basis for quite a few neurological problems.
This whole resting state sounds like it's setting the stage for the main event, the actual nerve impulse or action potential. How do they relate?
Perfect lead-in. Think of the resting potential like a spring that's been pulled back, holding potential energy. It's primed.
Ready to go.
Exactly. The action potential is the release of that spring. It's a very fast, very temporary flip in the membrane potential. It happens when a stimulus triggers sodium channels to suddenly open wide.
Ah, so sodium rushes in then.
Sodium rushes in, bringing all its positive charge, and this rapidly makes the inside of the neuron positive for a moment. That's called depolarization. That's the electrical signal, the impulse, that travels down the neuron. Then, very quickly, the neuron works to kick the sodium back out and let potassium flow out again to return to that negative resting state.
So the stable resting potential is absolutely essential. It's the baseline the neuron needs to return to be ready to fire again. It really is the foundation for communication.
Could have said it better. It's the necessary state of readiness.
And I guess it follows that things interfering with this resting state could cause serious trouble.
Absolutely. Many neurological diseases and also certain toxins actually work By targeting these ion channels or the sodium-potassium pump itself, they directly mess with the resting membrane potential, disrupting how neurons talk to each other.
Okay, so let's just recap quickly. This resting membrane potential, this difference in charge, mostly due to potassium leaking out, but actively maintained by that sodium-potassium pump constantly working, it's not really resting in a passive sense at
Not
all.
at all. It's an actively managed state of readiness.
Yeah, a state of readiness that lets our neurons fire signals and allows our entire nervous system to do its job.
Precisely. You could call it the silent workhorse of the nervous system. Always there, always ready.
So here's something to chew on, a final thought for you. Isn't it incredible how these seemingly tiny differences in where ions are located just across a microscopic cell membrane can have such massive consequences for our thoughts, feelings, actions, or overall health? What other questions does this raise for you about just how complex and finely tuned our cells are, and maybe how subtle imbalances could lead to bigger issues down the line? Definitely something to think about. Thanks for joining us for this deep dive.